Please continue the evolution part second in today's session. Please, sir.
Good morning, everyone.
Good morning, sir.
It's my pleasure that I had been allotted a task to deal with the process of organic evolution, which is one of the most fundamental aspect of knowing our presence on this earth. I take an opportunity to thank Dr. Kanade sir for involving me in this PhD course or program. And as per my and his wish, I had allotted the topic of evolution. I welcome you all. Dear friends, the organic evolution is the process which is not measurable with any standards. This is a change which is going to take place over the period of time, which has to be counted not in terms of years but millions of years. And so, whatever discussions have been made in the scientific world, these are the postulations, these are the assumptions, these are the theories, and they have been supported many of the times by some evidences. And for this purpose, yesterday we started our lecture with understanding the evolution and formation of the earth itself. That was the evolution of the physical world. And after that, we have took over the issue of origin of life, the biological origin, and the organic evolution. Almost all these three aspects, origin of earth, origin of life, and the mechanism of the process of organic evolution, it is still an enigma. We do know how it might have taken place, but we are not sure that this is the way only it has taken place. Obviously, when we try to understand something about the earth, we have to depend on the evidences which are present on the earth, at the same time the evidences which are present beyond the earth in our solar system and beyond that. Also, as far as the origin of life is concerned, still a very strong debate is going on how one should accept the origin of life on earth, whether on the earth itself the life was originated or it has originated somewhere else outside the earth and it has come back to us. And again, we are not having any concrete evidences for this. As far as the process of organic evolution is concerned, several theories they have been forwarded, and we have gone through these details. We have seen what Lamarck was willing to propose. We have seen the hypothesis which was put forward by Darwin and Wallace regarding the natural selection. And after that, we have seen the theory of de Vries, which was looked sound on mutation theory. But again, it has shown that it is incomplete in several aspects. And because of that, recently, a synthetic theory has been accepted. We had even suggested that environment and organism both are playing an interactive role among each other, and through this process of adaptivity at the morphological level and at the genetic level, the evolution must be taking place. And then, rather than studying the course of evolution, we try to understand how the speciation takes place, how a new species is generated, how from a population, a new population is derived, which is independent of its reproductive compatibility. And as far as the speciation is concerned, we have seen that there are several limitations which are present And these limitations play a very vital role in formation of the speciation. Also, we have gone ahead and we have seen that how the different disciplines, not only in the biology but in other sciences also, they are playing a very vital role to provide us a support, to provide us the evidence, to provide us some clues so that we can understand the path of organic evolution. And then we have seen that almost each and every branch of the science, including paleobotany, because they are providing the basic information, that is the fossil records or the life in the past. We have seen that the taxonomy, morphology, histology, cytology, ecology, and recently the two main aspects, that is chemotaxonomy or physiology and biochemistry, and the second is cytology. They have proved and they have paved a path for better and better and better understanding of this journey of organic evolution. Now, when we are talking about the organic evolution, one has to always keep in mind that whatever we see today, it is a result of what has been selected and what has been chosen in the process of evolution. Remember that these have not been made any form of favorism. Why this is so, no one could answer, no one can answer. So something was selected in the course of time and that persisted and it continued. And because of this continuation, because of this persistence, even though the environment has changed, the organisms that way they have not changed themselves. And because of that, we are seeing a great diversity in the flora and fauna of the world. That is nothing but it is the biodiversity. When we talk about the biodiversity, we have to think how the evolution of man might have taken place, human being might have taken place in the course of time, because we are the ones who are always asking the question where we have come from and how everything is generated. And when we started asking this question, we realized that this evolution has not taken place a single path, a straight roadmap. But we do know that in the past, more than 13 times, a mass extinction of life has taken place on the earth. So it was a catastrophic effect which has taken place. And this effect was such a time, or many of the times, such a high and adverse that more than 10 to 15, sometimes it is extended to 20 million years, the earth was complete in darkness. One has to believe that we are not talking about in terms of only days or months or years or hundreds of years or decades, but we are thinking of this process in the millions of years, thousands of years. And so when we go and when we try to understand what is the current situation, remember, the organic evolution is dynamic. Even today, the species which cannot tolerate, the species which are susceptible, the species which cannot modify, alter to the given environment, to their surrounding environment, they are supposed to be eradicated on the earth. They are supposed to be becoming extinct. And at the same time, there is a process of even formation of a new species. So on one hand, what has been formed, it is being removed. And on the other hand, what was not there in the earth, it is being created. And this is, you can say, a dogma of organic evolution. Now we have to go one step ahead. And today, in the second part of the evolution, we are going to discuss some of the issues which are related with the occurrence of angiosperms on the earth. Because the origin of man, the origin of human being, or the origin of Homo sapiens is beyond the scope of our syllabus. We are in the MSc botany, and that's why we should concentrate on the plants. And so rather than taking one group, one family, one order, or one genus, I feel that today's discussion should be concentrating, should be based only on the topic of what the organic evolution is saying about the dominance of angiosperms today. And this is the lecture which I am supposed to deliver today. And I hope again we'll be making a journey together and you will be enjoying this journey together. So let me share my screen with you so that we can go ahead with some of the interesting and important findings which have taken place in the course of time because of the active role which was played by the taxonomists, physiologists, and the people of the different disciplines. Now, when we are talking about the angiosperms, why the angiosperms are so dominant? That is the first question. Now, what is the definition of angiosperms? That itself is a question. Because in a very broad sense, we say that the angiosperm is a typical group which is showing certain characters. But let me tell you one thing, that within angiosperms, whatever characters we do take, we are finding the exceptions even today. And so to make a definition of angiosperm is rather a very difficult task. It may be a generalized definition that angiosperms are nothing but these are the plants which are producing the ovules which are enclosed in the ovary. That is the plants which are having the seeds which are present in the fruits. But dear friends, let me tell you that there are several genera where the seeds are born outside the fruit. That the fruit is not enclosed. The ovary is not enclosed, it is open. And the seeds are directly born on the stalk. And because of that, if at all we want to go ahead with this journey of angiosperms, one has to ask this question, why the angiosperms are so specific? So I will take just few minutes because I do know that you are knowing all these things together. We will make this comprehension of the angiosperms in a short overview and then we will go ahead. The angiosperms are distinct and they are dominant seed-bearing plants, as I told you, because the ovary is in, the ovules are enclosed in the ovary. On the present day, sharing the unique combination of characters. That is very important. Unique means obviously there are certain characters which are not found in other genera, other groups. The first obviously is the seed-bearing plants. Is it true? No. There are several genera, there are several orders where the plants are bearing the seeds directly, but they are having the covering of the fruit, but it is of open type. So the fruits are not completely enclosed, but they are open. So that's why their angiosperms are very typical. Now it is consisting, that is very true, it is consisting the vast and varied assemblage, biodiversity of more than almost 3 lakh species, which are spread almost in a cosmopolitan way all over the world. Again, out of these 3 lakh species, remember, almost more than 40% of the species, they are endemic and they are restricted to a specific region, ecological region. There's very few genera are there. It has been recorded that out of more than 32,000 genera, only 800 genera are present in all 5 continents. Whereas a large number of genera, they are restricted to the certain area, that's why they are endemic. And that's why in angiosperms, in other ways, they are not, even though broadly distributed, they are highly specific and restricted. They are considered to be geologically young in the sense that it is assumed that the angiosperms evolved somewhere about 100 million years back. On the contrary, the life of the earth is almost 4,500 years back, and the first plants originated somewhere about 3,000 billion years back. And the first land plants appeared somewhere about 600 million years back. So the first forests were present on the earth almost about 500 million years back. And that's why, from this point of view, the angiosperms, even though they are so widely distributed all the world, they are very young. In all the characters, gross and minute, external and internal, the sporophytic and gametophytic, the angiosperms, they show a great diversity of form as a result of adaptive specialization. That's very important because they are showing the adaptive specialization in response to the great climatic changes. So the moment the environment started changing, the angiosperms also showed the genetic plasticity, genetic elasticity, and they modified themselves. And they modified in such a vast way, vast potential, that we are having the maximum widest range of angiosperms in their diversity. And because of that, they are having this adaptive specialization. Because of that, obviously, what I told you, that angiosperms are maximally are endemic for the given areas. Some forms became highly elaborated, while the others became specialized by reduction, retrogression, and suppression. Now, what was said by the Lamarck, what was said by the natural selection by the Darwin, later it was found that it was incomplete. But the evolution is not taking place only in one direction, that is from the simplicity to complexity, or from a single cell to multicellular organism. But the evolution is also taking place with time from converting the complex species into simpler forms. So this simplicity is also an acquired character because of, again, a progressive evolution. So this can be described in the terms of reduction, retrogression, or suppression. And so angiosperms is the best example. The genera are the best example which are showing both the directions of evolution in one single genera, in one single family. So within the family, we find that some characters, they are highly evolved, and some characters, they are more evolved, but even though they are showing the simplicity because of the reduction in their characters. And because of that, the angiosperm is showing a genetic plasticity and environmental plasticity. Obviously, do you know that the sporophyte, that is the plant body, is a dominant life cycle, and it is having the root, stem, leaves, and many other similar characters, axillary characters. Angiosperms, they show the highest degree of perfection in vascular system. It is obviously xylem and vessels. But dear friends, as far as the xylems are concerned, up to gymnosperms, they are containing the sclerified cells, and these cells are called the tracheids. So the vascular xylem is containing the tracheids, and obviously xylem parenchyma, sclerenchyma, and so on and so forth. But basically, they are having a single unit, that is a tracheid, where the cells are closed on both the ends. On the contrary, in angiosperms only, the tracheids are replaced by the true vessels. There is no any tangential wall, and that's why the true vessels are present, and that becomes a very distinctive character of the angiosperms. Again, dear friends, let me tell you that in the renales, there are more than 14 families are present, which are angiospermic families, but they do not contain the vessels. But they do contain the tracheids. That is, the angiosperms are present without vessels, just like similar to the gymnosperms. Similarly, there's one character, that is the presence of occurrence of companion cell in the phloem. Remember, this is a very unique character. Companion cells, they are present in all angiosperms without any exception. But very interestingly, companion cells are also present in Nettles group of gymnosperms also. So companion cell is a specialized cell which is present near the sieve tube, and it is helping in providing the ATP, a force of energy, so that the solute material can be pushed, and that's why the translocation can take place. You know that translocation is bidirectional. And because of that, the companion cells are present in all angiosperms without any exception. But unfortunately, the companion cells are also present in gymnosperms also, that is in Nettles group. Organization of microsporophyll, that is stamens, and the megasporophyll, that is the carpel, together they form a structure which is known as a flower, which is typical only in the angiosperms. That is, in angiosperms, they bear the flower, which can be called as a complex bisexual cone. Such a type of bisexual cone is never in existence in all the groups. In pteridophytes, in gymnosperms, the cones are present. Male cone is separate, female cone is separate, but the cones are separate and they are born in a separate locations. Generally, the microspores are born in the lateral position and the carpex are born in the terminal position. On the contrary, angiosperms are the ones which are having a single specialized stem on which micro and megasporophiles are born together. And that's why this is called the flower. And remember, such a type of flower character is present only in the angiosperms. Again, in angiosperms, there are several exceptions are present. For example, in renales, there are several flowers which are spirocyclic and which are unisexual, where the male and female flowers are born at the separate locations. The total order Amentiferae, which was identified by the Eichler, you do know that which was the basis of, hello? Which was the basis of Eichler's system of classification, which suggested that Amentiferae is the group which is having the plants which are having the unisexual flowers. That is, the male flowers are separate and female flowers are separate. It means they are not containing a typical angiospermic flower. So they do have the flower, but the flowers are in the presence of, in the form of the unisexual flower, that is, the flowers are having the male characters and female characters separately. Very interestingly, there is one record of a fossil gymnosperm known as the Benititales. And this Benititales showed such a type of bisexual sporangiate cone. It was born laterally. And that's why Cycadoidea or Benititales is a unique gymnosperm. All otherwise, it was very similar to the Cycadales, but the only difference was that it was having a cone which was bisexual. It was having the microsporophiles and megasporophiles which were born on the same axis at the same location. So there are so many variations that I'm telling you. Then the presence of 4 microsporangia per microsporophils. It is microsporans or polysacs. Unfortunately, in gymnosperms and in pteridophytes, the number of microsporangia are many. There is a polysporangiate condition is found. Angiosperms, only one is there. Only a tetrasporangiate condition is present. The ovules are always enclosed in an ovary, which is the basic character, basal character of the megasporophiles. But again, this statement is having the several exceptions in angiosperms. The production of 2 kinds of spores, that is very true. We are finding that this heterospory nature, it has been developed right from the pteridophytes. So microspores, pollen grains and megaspores, that is the ovule, and obviously further embryo sac. So that's why the angiosperms are thus heterosporous, very similar to that of pteridophytes and obviously gymnosperms. The presence of single functional megaspore which is permanently retained within the nucellus or the megasporangium. Now remember, this is again a unique character of angiosperms, because in pteridophytes and in gymnosperms, the formation of a female gametophyte takes place because of the development of 4 megaspores. So the megaspore tetrad, which is formed after the reduction of or after the meiotic division by the megaspore mother cell, the 4 tetrads which are formed, they all take part in the formation of a female gametophyte in pteridophytes, pteridosperms as well as gymnosperms. However, in angiosperms, of this tetrad, only either the terminal one or the basal one, only 1 megaspore further develops into the female gametophyte, whereas the remaining 3, they degenerate. And that's why the single functional megaspore, again, it is a unique character of angiosperms. Adaptation of a flower to insect pollination, that is very true, that this is a character which has been acquired by the angiosperms, and because of that, these are helping in the form of a vector for cross-pollination. And the beauty of spread of angiosperms is because of this character. Unfortunately, again, the many advanced angiosperms, they are either hydrophilic or they are anophilic pollination. That is, they are depending on the wind or depending on the water. So the insects, it has been suggested and proved that the insects have evolved along with the development of the flower or formation of the flower. So the floral architecture, floral structure, and the insect development, they have taken place side by side. Obviously, pollination consists of transfer of pollen grains from one to another stigma. Unfortunately, such type of stigmatic character is a unique character in angiosperms, whereas in gymnosperms and pteridophytes, the style and stigma, they are totally absent. On the contrary, the pollen grains or the microspores, they are landing directly on the archegonia. So there is an archegonial tube and the pollen grains or microspores are landing in the archegonial tube, directly they are reaching to the egg. In angiosperms, unfortunately, there is a specialized structure of a style and stigma where the pollen grains are loaded, there they germinate, they produce the pollen tube, and this pollen tube carries the male gametes. The spore dimorphism is resulted in the production of endosporic gametophyte chicks, male and female. Obviously, that is very true. You know that male and female gametes are having the different sizes, structures. The extreme reduction in size, duration of existence, and complexity of the structure of endosporic gametophytes. That is obviously the structure of the pollen grain, which is containing only the two cells. One is a generative cell and another is a reproductive cell, or generative nuclei and secondary nuclei. So they are highly reduced. Even in case of female gametophyte, the female gametophyte is restricted in the form of only embryo sac, and then it is containing only one gamete. The archegonia are totally absent and they directly contain the egg. The male gametophyte has reached the limits of reduction. As I told you, they are only nucleate structures. It consists of the pollen grain producing the pollen tube containing the tube nucleus and two male gametes which are both functional. Sometimes the gametes are present only in the form of a nuclei also, and the male gametes are non-ciliated. In pteridophytes and in gymnosperms, the male gametes sometimes they are ciliated because they have to swim in the fluid, archegonial fluid, and they have to reach to the egg. Whereas in angiosperms, they are brought through the pollen tube. The female gametophyte lacks any extensive development of vegetative tissue. So there is no vegetative tissue at all, it is there. The embryo sac consists of three apparatus cells. Obviously, they are having one egg cell, the three antipodal cells, and the two polar nuclei, which are present at two different polars. Obviously, they are having one central secondary endosperm mother cell. So non-motile male gametes of the nuclei, they are carried bodily. So the gametes are not freed, but the gametes themselves, they are moving in the neighborhood of the egg apparatus through the pollen tube. The seed or the seeds, they remain enclosed in the ripened ovary, obviously, which is called as the fruit after fertilization. The phenomenon of double fertilization or the triploid or the triple fusion is a unique characteristic feature of angiosperms, and that is very correct character. You do know that in gymnosperms and pteridophytes, the endosperm is developed before fertilization. An endosperm is nothing but it is a part and parcel of the female gametophyte of the nucleus tissue. And because of that, in gymnosperms as well as in pteridophytes, the endosperm is haploid in structure because it is developed from the gametophytic plant. Whereas in case of angiosperms, the endosperm is always triploid and it is a forced fertilization process. So the two male gametes, they are exceptionally fertile, they are fertile and they are active, and one unites with the egg and another unites with the endosperm, and that's why it's called a double fertilization. Such a type of double fertilization is totally absent in gymnosperms as well as it is absent in pteridophytes. This triple fusion is again a unique character found in the gymnosperms, found in the angiosperms. The endosperm develops after obviously fertilization and it's diploid. Now the angiosperms, it's an ecological component. that the angiosperms are completely adapted to the life on land, water, or air. So obviously they are either terrestrial in a large quantity, they are aquatic also. There are several families, there are several genera which are developed in water, and arboreal means they are growing in the form of saprophytic form. Most of the orchids, the maximum, the most complicated evolved family is basically a saprophytic form. And so the angiosperms, they are evolved obviously for their ecological domain. Now, when we are talking about this, now let's come to the way with which the angiosperms might have evolved. Dear friends, we do know the classification of England and Mantle, Bentham and Hooker, even up to the Hutchinson also, and then to some extent Bessel. These classifications, they were based on two basic characters, that the angiosperms can be divided into two broad groups. One is dicots and another is monocots. Dicots are having certain characters and monocots are having certain characters. Now when we are talking in the PG level discussion, let me tell you that there are several dicots are present which are having the parallel leaf venation, which is a monocot character. There are several dicots are present, especially in the group Daphnes, where the seeds contain a single cotyledon, a single functional cotyledon. Now, there are several dicots are present which are having a scattered vascular bundle, which is very similar to that of monocotyledons. So, even though it is true, there are several dicots are present which are having a trimerous arrangement of the florals. Because we don't know that tetramerous or pentamerous arrangement is for the dicots. On the other side, we are knowing so many monocots which are bearing generally a reticulate venation. We are knowing the monocots which are having a secondary wood in their character. We are knowing the monocots where two cotyledons are present, even though they are placed in monocots, where one cotyledon is much larger and another is much more smaller and it is rudimentary. So these are very broad spectrum discussions. Again, there is one more view and approach, that is the position of the ovary. Because the position of the ovary is related with its location, its placement on the receptacle. Whether all floral parts they are present below the ovary and the ovary is present at the top, that is superior condition, that is ovary superior, hypogynous condition, whether this is a primitive character or whether the ovary is present at the base and all other floral parts are present at the top, that is the epigynous condition, it is a major question mark. And so when we are discussing, we have to keep in mind that as far as dicots are concerned, in a broad sense, we are having three major lines. One, whether the perianth is present or no, because many of the groups they are having the perianth, but others do not contain the perianth. The perianth is totally absent. Then if the perianth is present, then there is a presence of calyx and corolla are both are petaloid or sepaloid. That is whether the perianth is differentiated or not differentiated. Further, what is the position of the ovary? Whether the ovary is superior or whether the ovary is inferior. And then furthermore, whether the perianth is present in two or more whorls or the perianth is present in a single whorl. That is again a character of categorization of the angiosperms. Furthermore, again there is one distinction, that in angiosperms sometimes the floral parts they are not in the presence of, they are not produced in the whorls, but they are present in the spiral arrangement. So this arrangement is called a spirocyclic arrangement. So that is also a unique character. So when we are talking about the angiosperms, up to this level, up to Hutchinson, they were believing that angiosperms can be grouped in a broad sense, dicots and monocots on one side, and within dicots then the free petals or polypetalic condition and gamopetalic condition. They should be treated independently. And then within this group then we identified whether petals are free but the ori is superior or inferior. The petals are fused, ori is superior or inferior. For example, let me tell you one thing, that if you talk about the Bentham hooker, we do know the polypetally, which is having the three series, thalamiflory, disciflory, and caliciflory. In thalamiflory and disciflory, ori is superior, but in caliciflory, the ori is obviously inferior. Similarly, in the another group, gamopetally, in inferior, the ori is inferior, whereas in bicarpellary, the ori is superior. So the position of the ori was not considered as an important phylogenetic character. But later on it was identified that this is a very unique and important character, and the stress has to be given on how the evolution is taking place concerned with the position of the ori. So at the moment, after Bessey, who suggested that the angiosperms, they have not come from a common stock. Let me tell you one thing. What was suggested? The angiosperms, they are acquiring the characters because they have developed from different ancestors. And why they are showing the similarity is because of the parallel evolution. And once this concept was accepted, then it was realized that the angiosperms are basically, they are polyphyletic in their origin, that they are having many ancestors. And from these many ancestors, the present angiosperms, they are formed. And that's why we are getting a large diversity. Now, such a type of approach was accepted and it was put forward by the Bessey. It was followed by the Smith, it was followed by the Cronquist, it was followed by the Taktajan, it was followed by the Dalton, it was followed by the Thornpey. And in 1990s, then it was realized that actually the producing and giving the classification of the angiosperms, it is not a task of one man. Because the last man was Thorpe and Dalton, who tried to put all the angiosperms together. But later on, the different schools, they started working independently and together also. And they tried to provide a scene, they tried to provide a theory how the angiosperm might have evolved and what is the correlation of this angiosperm or different groups of within themselves. And this particular school, which was located in different universities, in different institutions, but they were working with a single goal and single task of arranging the classification or categorization of the angiosperms. This is called as an APG. That is angiospermic phylogenetic group or angiosperm phylogeny group. So remember, angiosperm phylogeny group, it is containing more than 32 schools, universities. They are working independently and they are proposing together. And that's why what we see today, from the present scenario, we have to see towards the angiosperms, how they are suggesting. So from their point of view, the lines of evolutions, as far as they are concerned, there are three basic lines of evolution of the angiosperms. The first line of angiosperms, it contains the Reynolds is considered to be the most primitive, as it was suggested by the Bessey in 18th century. It was accepted obviously by the Bentham and Hooker, and then it was followed by a school which was called the Hutchinson, Smith, Taktajan, Conquest, and many others. And they suggested that Reynolds is considered as the most primitive because Reynolds is the one which is having the characters like absence of xylem vessels. As I told you, that this is the Reynolds group which is having the xylem tracheids in their wood, not the xylem vessels, which is an advanced character. There are several families like Magnoliaceae, Winteraceae. Now, Alismataceae is from the monocotyledons. But Alismataceae again shows the absence of xylem vessels. And it is assumed that from the Reynolds only, the Alismataceae has given rise to the monocotyledons. That's why Alismataceae is important. Then the close family of Alismataceae is Trochodendroceae. And here, instead of xylem vessels, the tracheids are present. Then, as I told you, the spirocyclic arrangement of floral morphology. So here, it has been assumed that flower is bisexual, the carpels are many and free, the stamens are many and free, and they are protected by a perianth lobe or bracteal lobes, which are foliaceous, leafy, and they are protecting this, and they all are arranged in a spirocyclic manner. So it is not a flower as such by definition, but it is a bisexual cone, bisexual strobilus, and that's why they are being discussed from this terminology, and that's why they are having the analogous anatomy in compare to the gymnosperms and pteridosperms on other sides. The best example is the Zysandra in Magnoliaceae, which is having a triradiate pollen grains. That is, they are having a pollen tetrad, which is having the ridges, triradiate marking on its body. Remember, triradiate marking on the pollen grains is found, or the microspores is found, and it is a pteridophytic character. It is not an angiosperm character. So, Zysandra shows an occurrence of formation of a pollen tetrad. Now, binucleate pollen grains are released from the microsporangium in families like Magnoliaceae, Ranunculaceae, and Winteraceae. That is, remember, in other cases, the pollen grains, they are containing three nuclei. One is structural nucleus or structural cell, generative cell, and gametophytic cell. Because in case of these renales, the pollen grains are binucleate. They are containing only two cells. That is gametophytic cell or structural cell, and generative cell, which is going to give rise to the pollen tube. Which is going to give rise to the pollen gametes. So these are present in the families like Magnoliaceae, Ranunculaceae, and Winteraceae. So much discussion can be made on this line. Basically here there is one more line that in all these cases, you can see that ovary is superior. All these plants, basically they are woody plants, and they are containing obviously a typical floral character, generally born not in an inflorescence but an individual, single. So obviously this first lies, angiosperms further they evolved in the three lines of divergence as I told you, and the first line, it consists of the rhodioles, sarracenials, varietals, malveals, geranials, sapindales, rhamnels, and many others. Basically, these members are from the thalamiflory and disciflory, but the important character is that they are having the superior ovary. That is, they are having the hypogynous condition, ovary superior. Ultimately, in this first line, the climax has been achieved by the family Lamiaceae. That is, in Lamiaceae also the ovary is superior, but the flowers are most complicated. They are bilabiate, because in other cases the flowers are triradiate or they are radially symmetrical. They are pentamerous, whereas in Lamiaceae the flowers are in a single plane. The rhodioles and sarracenials, they have evolved parallelly from the renales. So again there is a divergence, separation of these two groups. Rhodioles is the one group and sarracenials is another group. Rhodioles group further developed itself into a woody habitat. For example, we have to go for the Myrtaceae. We have to go again and ahead with the other families like Rhizophoraceae and many others. Even the Solanaceae is included here. And on the other side, in sarracenials, this is the group where it is producing the herbaceous superior members like the family Brassicaceae. The family Brassicaceae, Geraniaceae, the family Leguminosae, that is Papilionaceae, herbaceous plants. And they have been developed parallelly in this way. And this was the clue which has been taken up by the Hutchinson to separate the records into woody plants and herbaceous plants. A definite relationship between the parietyls and rhodioles due to the presence of gynophore. Now you do know this character, that in the parietyls, the family Caparidaceae, it is showing the presence of gynophore basically, and also it shows the presence of androgynophore. The malveals are evolved through Bombycaceae, where the imbricate estimation is there, and where the stamens are many, and they are born in a many bundles. That is, polyadolphous condition is present, and furthermore, they are related to the geranials. Ultimately, the geranials, because they are containing the saponins in their body, they are related to sapindales. You know that sapindales are rich in glucosides, and later to rhamnels. Again, rhamnels are rich in glucosides, and also they contain the terpenes. The second line consists of campanules, roselles, myrtles, umbellales, and rubiales, where we will find that here the ovary is basically changing its location from superior position to inferior position. For example, in order roselles, you do know that the family Leguminosae, especially the family Papilionaceae, they are having the superior ovary, even though they have been placed in roselles. Whereas in all other members, like including myrtles and rosacea, etc., the ovary is inferior. So the ovary is slowly, it is pushing, it is being pushed downwards. In other words, the floral parts, they are pushed upwards. So it is not the position of the ovary which is being pushed, but it is the position of other floral parts, they are being raised up. And that's why the ovary is being protected. And so the hypogyny is considered the primitive character, whereas the epigyny is considered as an advanced character. The campanulales are the top of this line with the pappus calyx and high zygomorphy and the fusion. Campanula flower, you do know well. And these flowers, they are very allied and similar to that of a bicarpellity. That is Apocynaceae to Bignoniaceae and Tropilariaceae. They are supposed to be present here together. And then the myrtles show a range of epigyny. Sorry, I was talking about the Bignoniaceae and Tropilariaceae. No, I was supposed to talk with the Sapotaceae and Ebinaceae, which are having the inferior ovary. Now, myrtles show a range of epigyny to perigyny to epigyny, and also there is a presence of hypanthodium. That is a concave disc on which the floral parts are arranged. That is called the hypanthodium. So here the receptacle is enhanced. The umbellales with the epigyny and syncarpy, they are very closely related with the rubiales. The only difference is that in umbellales, obviously, the essential oils are present. In rubiales, the essential oils are absent. In umbellales, the floral parts are uneven and they are free. In rubiales, the floral parts, they are always fused and they are tetramerous in multiples of 4. So obviously there are umbel inflorescence, and also they show the presence of an advancement. So it is a sort of a phylogenetic advancement over the myrtles group. Obviously this is third line. I was talking about these two lines in the dicots now. In one case, we were talking about the ovary were superior, and in other there is the Reynolds group, which is having the from primitiveness to advancedness. So they started the families like Magnoliaceae on one end and ended with the family Lamiaceae and Verbinaceae, Acanthaceae on the other side. It is a group of sonales. The second line started with the inferior ovary. They started with the parietals and they ended with the campanulales and rubiales. And obviously the third line, it includes the lineals or orchideals. It shows the few resemblances with monocots, but they are having very common characters, and at the same time they show the parallel characters with that of dicots also. So both are showing the monocot as well as dicot characters together. The lilials differ from the Reynolds in number of floral parts and fusion of carpels. That is, in Reynolds, the floral part number is always more than 3. 4, 5, 6, 7, and so on and so forth. Because it is a cyclic sexual arrangement, a cyclic arrangement. Whereas in lilials, it is the flowers are always trimerous. So here the reduction is taking place. Obviously, fusion of the carpels, that is in Reynolds. Initially, the carpels are free, lateral carpels are united. In lilials also, initially, apocarpy, the carpels are free. We have to go to palmi, where the carpels are united. Again, remember, in palmi, the carpels are free, they are not fused. So ovary is trimerous, inferior and apocarpus. The family Iridaceae and Amarlydiceae are more advanced. Obviously, both are having the inferior ovary, and that's why they are placed in the epigyne. The Poaceae seems to be originated from Liliaceae. It is very close because the Liliaceae basically it is the movement of the plants from the aquatic habitat to land habitat. So many of the Liliaceae members they are present as a marshy plants and many are present as a land plants. Unfortunately, on the contrary, in Poaceae all the plants they are terrestrial plants. Poaceae family, that is grass family. However, the reduction in the number of floral parts, especially the perianth, reflects its advancement. Because in Poaceae, the structure of the flower is not only including the components of the flower, but it is including the components of the inflorescence also. So the bracts also becomes a part and parcel of the flower. The stalk of the inflorescence also is very important in flowering, in formation of floral architecture in Poaceae, and that's why the Poaceae is considered as the most advanced family among the angiosperms. One can talk only on the floral architecture independently in a separate lecture. It is such a way complicated and showing advancements and combinations of different things together. Orchidaceae, due to high zygomorphy and variety infusion and entomophily, is the most advanced among the monocotyledons. Because Orchidaceae is having the floral complexity, it is having the pollination complexity, it is having the dispersal complexity, and it is also showing the ecological complexity. Because the presence of velamen tissue, it's a very unique character where the roots are becoming photosynthetic and they are performing the function of photosynthesis. Even the roots are having the capability to absorb the moisture from the air, and that's why from genetic point of view the Orchidaceae is much more advanced than the Poaceae. Even though grasses are spread over a large number, actually Orchidaceae is more advanced. So the next question one has to ask when we are talking about the evolution of angiosperms is, are the angiosperms monophyletic or polyphyletic? That is, they originated from a common single ancestor, or they evolved independently from different ancestral groups. Are they polyphyletic, that is descendants derived from a single ancestor, or polyphyletic, that is group does not have the common ancestor? Angiosperms are a natural group with unique characters, vessels, combined cambium cells. Obviously, they are having the bisexual architecture, and the ovules are present enclosed and double fertilization, which unique character which we have discussed. Now, the two trains, the initial train was the monophyletic origin. There is Dalton, Doyle, and Donegay, and Fregri, and Takhtajan. They considered that angiosperms show certain unique characters. They show the vessels, they show the bisexual strobilus, they show the double fertilization, and they show post-fertilization endosperm development. And that's why it should be considered as a monophyletic origin. So because of this, it shows a remarkable consistency in presence of sieve tubes, endothelial wall layers of the anther, double fertilization, triploid endosperm, and also occurrence of monosulcate pollen. That is, pollen grains are having a single seizure, single opening through which the pollen tube is coming out. That is called single monosulcate. However, unfortunately, such a type of single ancestral fossil evidence is not available with us. As per the polyphyletic origin is concerned, this was proposed by the Conquest, Hughes, James, Crasilau, Mayhew, and many others. And now it is also been pursued by the APG group. It is argued that the dicots and monocots, they have originated from different stocks. Not only that, but they have originated from different times. So even their period of origin of ancestry also is not same. But unfortunately, or whatever it may be, they have attained the present status through the parallel or convergent evolution. Convergent means similar. They have the present status achieved because of the parallel evolution, common characters which are acquired, and as well as by the similar evolution. So this is also supported by the fossil records of variety of peria and nature of carpels in both dicots and monocots. That is, the dicots are separate and monocots are separate, and this particular floral architecture, wood anatomy, it is consistently being recorded even in the fossils. That's why monocots and dicots are separate, and that's why it is assumed that they must be having the polyphyletic origin. The primitive orders of both, there, however, does not show any close relationship in their characters. It means even though the characters are present in the angiosperms, in ancestral forms, there is no any close relation, there is no any meager, there is no any mixing of these characters together. So ultimately, the fossil record suggests that the angiosperms as a group are monophyletic. The whole angiosperms, they evolved from a single common ancestral group as such, and their families and natural groups which are developed further, they are polyphyletic. Means once the basic divergence took place, this branching took place further independently, and so today's families are more natural and they are coming from their ancestral stock, not from the common ancestral stock. Recent phylogenetic analysis using the nuclear, mitochondrial and plastid gene sequence claim that monocots and eudicots, they are polyphyletic, they are monophyletic. That is, these two groups, they are coming from the common ancestor, but apart from the eudicots, the other dicots, that is bracketed dicots and primitive dicot or advanced dicots, they are derived from the different groups. The angiosperms as a whole were found to be monophyletic to the exclusion of gymnosperms. It means that angiosperms, they have evolved independently from some common ancestor, and angiosperms have not evolved from any common ancestor from the gymnosperms. That is, from genetic sequence, it is very clear that gymnosperms have evolved independently apart from the angiosperms. There is a major question which has been asked, and it is debated still today, and that is regarding how the monocots evolved. What happened? Why the monocots are having their unique characters and these characters are separate than dicots? Now, it has been proved now that the monocots are more evolved within angiosperms, and monocots have evolved somewhere from a dicotyledonous ancestral stock. So there was a common ancestor, and in the early branching, one branch developed into a dicot group and another branch developed independently into the monocot group. To explain, several hypotheses they have been proposed. Obviously, each and everyone is having more objections than the common characters. So Engler and associates suggested that isoetales group and ophioglossales group of pteridophytes, which are primitive on one side, aquatic on other side, and they are having a specialized gametophytic nature, and a few other pteridophytes as ancestors of monocots, because it is now accepted that the primitive monocots were aquatic plants, and from these aquatic plants the land monocots they have evolved. This is because the aquatic herbaceous habit of the monocots, that is the alismatici group, and that monocots are more primitive than the dicots. It was a suggestion. Now this has been not accepted, and now it has been claimed that dicots are more primitive than monocots. Families like Menispermaceae, Ranunculaceae, Largizapalensis, Barbadiaceae, they have been treated as a possible ancestors because both are having the herbaceous habit and monopulpate pollen. They are having a single radiate mark on their body, and which is considered as a primitive character. Now, the same character is also exhibited by the family Alismataceae, the family Aponogetonacea, and the family obviously Nasadaceae of the monocots. And as these are compared together, it is stated that from these primitive herbaceous dicots, the angiosperm might be what? Cartagen, Conquest, Arbor, Hellier, etc., they have the opinion that some ancient or extinct vessel-less herbaceous plants with the Apocarpus carpel must be a common ancestor, which will be having again a monopulpate pollen grain resembling the modern Nymphaeus group. So obviously you know that Nymphaeus come under the Rhenales group, and it is a herbaceous aquatic dicot as a probable ancestor. And that's why Nymphaeus is very strongly compared with the alismatales of monocots. This is because the vessels originated independently in the both. There is an analogy of the vessel development is totally separate in both Nymphaeus as well as in Alismataceae. So this group has captured and closed vascular bundles, very similar to that a monocot character. They are having the reduced primary root similar to the monocots again. They are having the apocarpus ovary, a primitive character, with a diffuse placentations. That is, many ovules are born on the receptacle. It is not a single placenta, but the whole placenta becomes a receptacle, and on this receptacle, the ovules are embedded. But obviously, there is a difference in the number of cotyledons. Because in Nymphaeaceae, we are having the two cotyledons, and in others, in monocots, we are having a single cotyledon. Also, the aquatic herbaceous habit is a strong issue of debate, suggesting that probably the more primitive and less reduced herbaceous dicot of Nymphaean type might be a probable claim. Now remember, when we are discussing the dicots and monocots and the habit, it has been proved that initially the dicots were terrestrial and they acquired the aquatic habitat. That is, the aqueous habitat or aquatic habitat is an advanced character in dicots. Whereas in monocots, however, the aquatic habitat is a primitive character and the terrestrial habit is an advanced character. So exactly it is reverse. Dicots evolved on land and they entered into the water. Monocots evolved in the water and they moved toward the land. There is a basic difference of ecological divergence as per dicots and monocots are concerned. Berger, very interestingly, he proposed that among the order there is one family, Chloranthaceae. Actually, this member is generally found in the Indonesia and Australia and New Guinea and other areas, which is showing a trimerous flowers. It is very close to monocots as it is having a single flower, having a single bract, only two stamens and a single adaxial pistil. So this is very similar to Lactoridaceae, Sauroraceae and Potamyotanaceae and Alismatales of the monocots. So it was a very interesting comparison which was made by the Berger, but unfortunately this could not get much more gain and support later on. But definitely this claim has to be studied furthermore because it is one of the interesting claims. Until recently, it was assumed that the anthophyte origin, that is from the phalerogams, whereby the nettles are having the common characters, the nettles are sisters to angiosperm. That is, they are having a common origin because many are having very common characters, and this is because of the parallel evolution. And that's why this was a popular concept. However, the inability of the molecular data to resolve this relationship has added not to solve the problem, but to create the more conflict. The molecular data tree topology is now, that is the cytological analysis and the proteins isoenzyme analysis. It is now pointing towards gymnosperms are monophyletic, that is gymnosperms derived from a single ancestor, but they are not derived from the angiosperms, but they are sisters to angiosperms. Means gymnosperms on the other hand and angiosperms, they are having the common origin. And this relationship is born out by the genes derived from the nuclear or plastid or mitochondrial genes. And so, as far as this whole process of evolution is concerned, somewhere about 200 years back, the Darwin has said that still the origin of angiosperms remains a mystery, a problem, a question, as it was rightly pointed out by Darwin almost 100 years ago by suggesting that angiosperms have an abominable theory. Abominable means unexpected, shocking. So it is not having any clue at all. It is not having any relationship at all. And all of a sudden we are finding the angiosperms everywhere. And in this way, when we try to see the evolution within the angiosperms, we are still in the conflict. So to go ahead with some of the ideas which have been proposed related with the origin of angiosperms and its correlationship, I will take just like 5 minutes extra and go ahead with some discussions. So what was the probable time of the origin of angiosperms? As per the fossil record concerned, let me tell you one thing, that I have to go back and show some slides. Okay, it was not here in this slide. Okay. Early Cretaceous period, that is obviously nothing but it is the end of Mesozoic era. And this Cretaceous period is almost 135 to 125 million years back. And late Cretaceous period, about 100 million years back. Remember, from the Cretaceous period onwards, the paleobotanists, they started getting the confirmed form genera, confirmed form material, angiospermic material from the fossil records. We started gathering the petrifactions, leaf impressions, root impressions, seed impressions, pollen grains, even the petrified, silicified wood of the monocots. So all this material was abundant in the early Cretaceous and late Cretaceous. And however, before that, so remember, Cretaceous period is nothing but it is the end part of Mesozoic period. And later, some scientists, they suggested that if at all this period is showing the advanced families, one has to trace the angiospermic origin much more earlier. And this went back to Permian period, that is about somewhere about Paleozoic period. So the Paleozoic period was present about 280 million years ago. 280 million. So for determination of first traces, we have to depend obviously on the fossil record. Unfortunately, the paleobotanical studies, they are not able to solve the mystery because they are not going to give the concrete record. Unfortunately, whatever the records are available, these records also, they are very close to the advanced families of angiosperms. They are not the primitive families, they are of advanced families. And that's why this record is not that much significant. As far as the late Cretaceous period is concerned, a vast and rapid spread of angiosperms is one of the most important events in the history of time. The whole process, it almost took 25 million years to generate and to establish the angiosperms on Earth. And this is about the half of the period of the Cretaceous region. And this is considered neither the fast or slow movement for the progress of organic evolution of angiosperms. As far as the reports are supported by the fossil records. Obviously, in the Permian period, by opinion of many authors, the angiosperms undoubtedly originated much more earlier than Cretaceous period. So even though the records are available in 125 million years back, we have to find their trace much more earlier and somewhere in the Permian period. That is at the end of Paleozoic period. Why? Because it has been proved cytologically, biochemically, and also anatomically that gymnosperms and angiosperms, they were having the common characters. So if at all the Jurassic period or if at all the Mesozoic period is having the large number of gymnosperms, we have to go much more earlier than this era. So this is a sort of a graph. Now you can see in the Mesozoic era, you can see the end, that is the Cretaceous period, and see this last right line where the angiosperms are written. So the record of the angiosperms, it extends up to Triassic period. However, the Permian period is the one where the Paleozoic period is ending. And so somewhere here, we have to find out the probable time where the angiosperm might have evolved. As far as the probable place is concerned, I'm not talking about the place of origin because it is a question mark. Almost each and every region, I'm talking about the locality now, zone of the earth, it has been considered as the origin of angiosperms. But before discussing this issue, we have to keep in mind that today's 5 continents, today's land mass, which is separated by the sea or ocean, it was not as such, it was not similar to what was present almost 100 million years back. Why? Because the records show that 100 million years back, all the angiosperms, all the land, it was present together, and that land was called as a Pangea. And in this Pangea Gondwana region, it was a continuous landmark which broke down and spread into the space. So the probable place is to be sought through the fossil evidences, through distribution, spread, and diversity pattern, the abundance of primitive species and their geographical distribution, and evidences through the developmental botany, histology, cytology, which are providing the claim. So in this discussion, the 2 important issues are the role of phytogeography, that is, in which location we are finding the fossil records, and second, the phylogenetic relationship. So what the fossil records say, and what is the present flora of that area. These are to be compared to answer this question. Now, as I told you, see the image that all the 5 continents which are present today, they were not separated. On the contrary, all the land masses, they were present together. And this was called as a Pangea. So this is the map of the land in early Cretaceous period, 130. But here something happened, and because of that, these lands, they split and they started moving. And see this late Cretaceous period now, which is 90 million years ago. And now here you can see these land masses, they are splitting, but even though they are connected to each other. And you can see in the very near to Antarctica, Antarctica Chidari, near Antarctica, we are having these 4 land masses together. They are attached to each other. South America, Africa, India, and Australia. So all are connected together. That's why it's a very interesting thing. And see the beauty. This is the present day. What we are having today, Antarctica is present in the southern region, whereas all other land mass is present in the northern hemisphere. Australia, Africa, and South America, they are completely separate from one another. On the contrary, very interestingly, India has moved with the fastest speed and fastest distance. So you can see at one corner India has reached, and it has then not only reached, but it has pushed the Asian region or Asiatic region, and because of that, there was a formation of Himalaya. So, as far as the probable place is concerned, if not the birthplace, we have to find where the primitive families are gathered together and whether these families are giving us some clue. So, many true primitive families or genera are present in the what we know as a Pacific Rim, and this region is also showing the advanced angiosperms. For example, you can see this region now. So this is the image of Earth in the present context. So what we see at the center, हा मध्ये जो पूर्णपणे पांढरा भाग दिसतोय हा सगळा पॅसिफिक समुद्र आहे, ज्याला आपण प्रशांत महासागर म्हणतो. And see, many primitive families, they are present around this rim of the Pacific region. So what it indicates? It indicates that this is the region where from the diversification of the angiosperms began. So Valentine proposed a species pump hypothesis, very interestingly, and here he suggested that a rapid and active speciation or formation of species takes place in the marginal areas of the deserts. That is, it is the area of unfavorable and favorable conditions. Generally, because of the stress condition in unfavorable regions, there is a genetic plasticity and elasticity. Because of that, new species are formed, and these new species, once they are formed, they are pushed into the favorable regions. So newer species are pumped into stable communities of tropical forests. And so angiosperms, in all probabilities, they might have developed into the semi-arid situations or on the borders of the deserts. If this is the fact, then to justify the speciation theory, we have to go one step back and study the geography of the world and try to find out where are the favorable condition regions and where are the desert conditions, which are creating a marked seasonal drought and also a short distance or short season for favorable condition for the formation of fruits and seeds. Now, this is what is known as a phytogeographical map of the world. And here you can see the dark green patches, they are of the tropical rainforest. Very interestingly, if you see the India, towards the eastern side, we are having the dark green, that is the region of Assam, Brahmadesh, Indonesia, Java, Sumatra, ज्या भागामधून मसाल्याचे पदार्थ आले असं आपण म्हणतो. And Assam, we are having the maximum rainforest, tropical rainforest. तुम्हाला सगळ्यांना माहिती आहे की चेरापुंजीमध्ये सगळ्यात जास्त पाऊस पडतो. Very interestingly, you can see that the desert also is ending in the India. म्हणजे भारताच्या राजस्थानमध्ये वाळवंट आहे, आपण जे राजस्थानचं वाळवंट म्हणतो, आणि त्याच भारतामध्ये हिमाचल प्रदेशमध्ये फक्त 400 किलोमीटर पलीकडे गेलं की त्या ठिकाणी जंगल आहे. So this is the area which is giving us an idea, a clue that the deserts and the tropical areas or the environmental richness is present side by side. To make the things more clear, see this zone. But this is the zone of distribution of deserts in the world. Now you can see the equator and the Tropic of Cancer, म्हणजे जे आपण कर्कवृत्त म्हणतो, म्हणजे विषुववृत्त आणि कर्कवृत्त या दोघांच्या मध्ये, आणि त्याचबरोबर खाली ट्रॉपिक ऑफ कॅप्रिकॉर्न, कॅप्रिकॉर्न म्हणजे मकरवृत्त, याच्यामध्ये सगळ्यात जास्त डेझर्ट्स आहेत, याच्यामध्ये सगळ्यात जास्त वाळवंट आहेत. मग जी वाळवंट याच्यापेक्षा वरती आहेत, दोज डेझर्ट्स विच आर प्रेझेंट बियॉन्ड दिस, दे आर कॉल्ड द कोल्ड डेझर्ट्स, and the deserts which are present near the equator, they are called the hot deserts. So the Sahara and the deserts which are present in the Mediterranean region, म्हणजे इराक आणि इराण त्यातलं वाळवंट, अरब देशातली वाळवंट, and in India obviously the Rajasthan, all these are present in this region. But this India is also having the Himalaya. So obviously on other side, if we come back, you can see we are having the tropical forest also. And because of that, according to the Stebbins, which proposed a museum concept, The tropical rainforests of this region serve as the museums because what they do, whenever the species are formed, these species they are pumped in this tropical belt and they are preserved. And that's why in all tropical and subtropical rainforest, we are finding the primitive family also and we are finding the advanced family also. म्हणजे ज्याप्रमाणे आपल्याकडं प्राणी संग्रहालयामध्ये सगळेच पक्षी असतात आणि प्राणी संग्रहालयामध्ये सगळेच प्राणी असतात एकाच जागी. तिथं सुसर पण सापडते आणि हत्ती पण दिसतो आणि वाघ पण असतो आणि हरिण पण असतं. सो दॅट्स वाय इट इज ऍक्टिंग एज अ म्युझियम. Maximum environmental challenge occurs on the margins of equatorial zone like the borders of deserts where the new species are originated due to the stress condition in environment. These new species, that is, I'm talking about the speciation now. These new species, then they are thrown in favorable climate to establish, and these are further preserved in tropical rainforest. Dear friends, in yesterday's discussion, we were discussing about the process of speciation. And in process of speciation, we have seen that there is a spatial barriers, that is geographical barriers, which are giving rise or which are actually the major components because of which the new species are formed. So this region and this discussion justifies the cytological evidence for the formation of new species. As far as probable ancestors are concerned, right from the algae, including chlorella, huge details, almost each and every group has been discussed. I'm not going to give you the details about this because you are already knowing this. The nature of probable ancestors of angiosperms is another problematic issue. Some morphologists have sought their possible ancestors among the seed plants, the Carboniferous and the Permian period, that is, they have seeked their ancestry at this region somewhere about 200-250 million years ago, and some of the well-known plant groups have been assumed to be particularly closely associated with angiosperms. So also the ancestor stocks is to be discussed by keeping some common closeness lines. Now, I'm just giving you this chart. I'm not going to discuss this in detail. So, which are the probable ancestors of angiosperms? Nutels, obviously, they are the present day advanced gymnosperms like Nutum, which are having several characters similar to that of angiosperms. Obviously, they have been compared to coniferals, where the cones are unisexual and male flowers are separate, female cones are separate. They are compared with the Amentiferae, that is Eichler's claim. Benitatails, as I told you, that this was the group of ancient extinct gymnosperms. This group was present in Triassic period. That is, this was the group which was having the bisexual strobilus, and they belong to the Cycadales group. It is also called a Cycadoidea or Anthostrobilus group. This was also discussed very widely by Arbour and Parking and many others. Then Chitonials group. Now the Chitonials was a pteridophytic extinct group which was present in Permian period, Carboniferous and Permian period. And in Chitonials, actually this was a pteridophytic group, but here it was showing the seed formation where there was only one central megaspore which was functional and other megaspores which were present, or not megaspore, megasporangia. The other megasporangia, they became sterile and they protected the central one. So that's why this was a very interesting record which was known as the Chitonials, where the seed formation was seen in pteridophytes. This is also compared with the angiosperms. Obviously, the another group is Pentoxylales, where it was very similar to that of today's Magnoliaceae and Winteriaceae group, where the wood was very similar to the Magnoliaceae, but it was having the tracheids obviously. But the leaves, they were very similar to that of leaves of leaves of Ranunculus members. So it's a Pentoxylales group. This group was also having the unisexual strobili. And then the most interestingly, the pteridosperms. because obviously this was the group which evolved from the pteridophytes, and this was the link between the pteridophytes on one side and gymnosperms and angiosperms on the other side, because these were the pteridophytic groups which were bearing the seeds. They were having the seeds on their body. So the female megasporophils, they were bearing the seeds, and obviously they were having the endosporic development, and because of that the pteridosperms, they are very highly developed. This Stachyspory and Phylospermy. This is the discussion which has been made by the Harper. It is related with the abaxial and adaxial position of the leaf, because it has been suggested that microsporangia, they are born on the abaxial side of the leaf. That is, they are born on the upper surface of the leaf, and which is very similar to that of pteridophytes. But the adaxial surface or lower surface is a characteristic of the cycadales. And that's why these two are compared together. As I told you, isoetales and monocotyledons, because both are aquatic, both are having the specific homosporous type of spore element, and that's why they are compared. Very interestingly, there was one theory which was proposed by the Durian, because Durio is an angiospermic genus which is present in New Guinea, which is present in Indonesia, which is present in allied areas, and it belongs to the family Bombaceae of the Rennes group. And this Durio genus, this is Durio gibbethinus. This is the group which has been compared with obviously the Pentoxylins once upon a time. So it's a very interesting theory. So many theories have been proposed, but still so far, not a single group is having very concrete conclusions so that we can say that this is the possible and probable group wherefrom the angiosperms might have originated. Obviously, in conclusion, it has been summed up, it has to be summed up, as the Pant and Kirwai have said in their book, that in spite of these heroic attempts to solve the problem from the different angles, there is no convincing evidence in support of either the accepted time, accepted place, and accepted nature of the ancestral stock or the place of origin of angiosperms. And what he has suggested, that today's primitive angiosperms, they themselves are advanced because they are acquiring, they are showing several certain advanced characters which it was not expected that their ancestors should have. So when we are having a broad spectrum of angiosperms today, within angiosperms we find that some families, they are highly advanced, some are moderately advanced, and some are very less advanced, and then they are showing certain primitive characters also. So it is a sort of an assemblage that the group Rennes, the group Amentepheri, or even in monocots, this is the group which is having several primitive characters and several common advanced characters. So it's an assemblage. All these plants, they are not reflecting, they are not showing all the primitive characters together. And that's why today's primitive angiosperm present families, they are not primitive angiosperms. From these, the other angiosperms not evolved. We have to find again one independent ancestral group. And for that purpose, we have to rely on the studies which are supposed to come from the different disciplines of botany. Obviously, to give a concrete claim that how the angiosperms originated, how the evolution in angiosperms took place, how the gymnosperms were separated at the beginning, how the monocotyledons were separated later, and how the hypogyny and epigyny generated, how from the spirocyclic arrangement, the cyclic arrangement took place, and why today angiosperms are showing the greatest ecological diversity. If at all we want to discuss, dear friends, as you are the students of PhD, it is a humble request, it's a need of time, Let's study and go ahead with a comparative account of taxonomy, of physiology, biochemistry, chemotaxonomy, histology, cytology, genetic studies, fossil records, serological records, and obviously the biochemical records of DNA, RNA, and proteins. Because these records, they are going to give us a clue, they are going to give us a light with which the darkened and hidden path of origin of angiosperms can be highlighted in coming years. With this, I complete my lecture series. I was given the topic of evolution to discuss. I separated them into two lectures. Yesterday we have discussed the process and progress of organic evolution, and today we have discussed the organic evolution in context to the angiosperms. I hope that I have come to your satisfaction. Even though I have tried to touch several areas, you have to go ahead. You have to read many books. You have to read several reference books. You have to read the monographs. You have to go through the online literature, study them well, prepare own notes, and then only present ahead or go ahead for presenting at the time of evaluation. Dear friends, to conclude, let me tell you that angiosperms is most challenging to study for the present, to study for the future, and to study for its past. Wish you all the best. To conclude, I thank Dr. Kanade for giving me an opportunity to be with you. I thank Dr. Sawan sir, head of the Department of Botany, for giving me and selecting me for this topic, and as he felt that I can give some light or some justice to this topic. So I thank both of them, and also I thank all of you for patient listening. Also, you will be getting the notes of my record. You will be getting this PPT in the form of record and in the form of a YouTube presentation to you in the due time. I wish you all the best for your future. Stay at home, stay safe, take care, study well. Wish you all the best. Thank you. Have a great time.